Okay, students, welcome back. This is part 2 of chapter 16. We're trying to figure out how DNA replicates. In part 1, we sorted out the structure of DNA, three-dimensional shape. Part 2 is all about how DNA replicates, and let's check it out. Nothing else exciting happening. So let's figure out DNA replication. So remember, DNA is double-stranded. It's two strands run in anti-parallel direction to each other. And DNA replication has to be accurate, right? Accurate, because you don't want to make a mistake in your genetic material. You don't want to pass along a mutation. So one of the things about DNA replication is it has to be very accurate. A second thing is it has to be kind of speedy. Think of human cells. Each human cell has 46 molecules of DNA, and on those molecules are roughly 3 billion base pairs. That base pair means a nucleotide, a pair of nucleotides. That's a lot of DNA that needs to be replicated if a cell wants to divide, right? We're thinking of a cell going through mitosis or going through meiosis. Before that cell can divide, it has to replicate all of its DNA. Has to do it quickly, has to do it accurately. So how is this achieved? First of all, when we look at replication, we say, where does it start? Where does DNA replicate? Well, in eukaryotes, like our cells, the DNA replication starts in many different spots along the way. Along the length of eukaryotic DNA are many regions where replication begins. These regions are called origin sites. Oops, I need to get myself organized here. Let's do red. These regions are called origin sites. Okay? Let's draw a picture of this in a second here. In prokaryotes, there's only one origin of replication. Remember, prokaryotes are much smaller. Their DNA is circular. Okay, they have one circular chromosome that's double-stranded DNA. And then they might have one spot somewhere on this circular chromosome. That's where DNA replication is going to start. But let's look at eukaryotes a second. So with eukaryotes, we have, a very long double-stranded molecule of DNA. If you want to put in the 5' to 3' and 3' to 5' designations, let's say this is one molecule of DNA. At different places along this molecule will be an origin site, a specific sequence of nucleotides where replication starts. Let's just say there are 3 of them in our molecule of DNA, but there would be many more. And at that replication site, DNA replication is going to go in both directions. So you're going to start replicating both directions from that origin site. Notice how fast that's going to be then, because we're replicating, starting at different parts along the chromosome, and we're replicating both directions. So what happens then, it goes something like this. If you've got at a replication site, you have the double-stranded DNA molecule kind of unwind from itself and form a bubble. We call it a replication bubble, actually. So at the origin site, the double-stranded DNA starts to unwind from itself, and then replication begins in both directions, and a new molecule of DNA starts growing, starts growing this way. So replication is going toward the left there at this fork. We call that little spot a fork, a DNA replication fork. And DNA replication starts going the other direction as well. So DNA is being replicated both directions at the fork, and pretty soon you get DNA replicating both directions, along each fork. And so we get rapid replication of DNA. And pretty soon we end up with 2 molecules of DNA that are each made up of one old strand, the red strand, and one new strand of DNA. Now all we have to do is sort out how this happens. Here's another picture of the origins of replication in eukaryotes, and you see it's pretty much what I drew as well, but you can see that the language that we'll use sometimes is a replication bubble and a replication fork. That's like the kind of juncture, right where replication is happening. Okay. All right. So how is replication achieved? That's the question. So let's see what I have. Okay. I'm just trying to figure out what I have in my notes. The first thing that has to happen is there is a protein called an initiator protein that finds the origin site and binds to it and kind of calls in all of the replication machinery. Hey, come here. This is where replication is going to happen, all right? Because we're going to need some enzymes. We're going to need some help to replicate DNA. We're going to need a lot of, and you can see, look at all the enzymes that we're seeing here. I'm going to change my color, I think, to, it's not letting me, to like a blue. Okay. After the initiator protein shows up at the origin site, the next, there are a couple other things that happen. One of them is that helicases then unwind the double-stranded DNA. So helicases unwind the double-stranded DNA at the origin site. We have to open it up so that it can be replicated, right? And once the helicase unwinds the DNA just wants to fold back up on itself. To hold the double-stranded DNA, to hold the DNA in an unwound position, there are things called SSB proteins, single-stranded binding proteins, that keeps the double-stranded DNA apart, right? So that keeps the SSBs, or single-stranded binding proteins, bind to the DNA and keep double-stranded DNA unwound. We need to have it unwound in order to replicate it, okay? That keeps the double-stranded, that's what DS stands for there, DNA unwound. So here's a picture, and we'll come back to these other things, but here's a picture of a piece of DNA and an origin site. So here's our origin site. Oops, I'm trying to draw it on here, and it's not letting me use the top part of our, like here is the origin site. And at this location, we've got a bubble. Notice that the double-stranded DNA is running 5' all the way over to 3', and the opposite or other strand is running anti-parallel to it. On one of these strands, I've put a couple of nucleotides, ATT, CCG. Okay. And what we've got so far is a slightly unwound double-stranded DNA, and I'm going to put into place a helicase enzyme. You should label it helicase. I'm just going to label it with an H because I can't write all those words out. The helicase enzyme has bound and now is unwinding that double-stranded DNA, so we're getting a big bubble. Then there are single-stranded binding proteins. Maybe do these in a different color. I didn't. That bind to the single-stranded DNA molecule. I'm going to put a couple of them in here. It's not liking me at all. I didn't do it. I don't know why. There we go. There we go. Single-stranded binding proteins bind to the single strands of DNA and hold them unwound so that we can replicate the DNA. All right. As we're unwound, and then down here we've got a bigger picture of the bubble, right? So I could put in, these A, T, T, C, C, G, if we wanted to. Okay. Now, what happens when we unwind DNA at this location is that we start to uncoil it. What happens to the DNA kind of upstream from this bubble? We're unwinding it here, and what happens like over here, or what happens over here? As you unwind one part of double-stranded DNA, what do you think happens to the parts upstream? They get hyperwound. They get overwound. It's a little bit like, when you're pulling out your Christmas lights and you start to pull apart a couple of Christmas lights because you haven't put them away very well. This happened to my dad every year when I was a kid, and he'd get really angry. And he'd start pulling at the Christmas tree lights, right, to unwind them, to separate them. He'd be like, yeah, this is working pretty well for a while. You can kind of unwind them for a while, but what happens upstream? Well, you got a disaster, right? The rest of the chain gets all messed up. That's what happens with DNA too, except there are enzymes called topoisomerases. Add a topoisomerase, a big guy. We're going to call that the topoisomerase. Topoisomerases will bind to the double-stranded DNA upstream of our replication bubble, and they will unwind. Well, basically, they will cut the DNA, unwind it, and put it back together again. They will, what do I have for my language here? I think I wrote something like this. Topoisomerases relieve the twisting DNA molecule by clipping and rejoining DNA. So if you want to put that in here, topoisomerase enzymes relieve the twisting DNA molecule by clipping and unwinding or rejoining the DNA strands. All right. Now we just are ready to go. All we have to do is build DNA, right? We've got the DNA the unwound. We've got it held in its unwound position. We've dealt with some twisting DNA upstream, and we're relieving that. Now we just need to build DNA, but we're not quite ready to, because the enzyme that builds new DNA can't start from scratch. So if I wanted to build new DNA, and I just wanted to just start building it, maybe I need to do something here. Maybe I need to write in, hold it here, maybe I need to draw in the, why is this not liking me? I really want to change the color. I really, really, really want to change the color. And it's not liking that. Oh, well, that worked. Okay, so I don't know if I wanted to change it anyway, but maybe I want to write in the complementary strand of DNA that matches ATT-CCG, but I'm going to put it up here, okay, just so we know. T-A-A-G-G-C, right? Okay, so if you want to build new DNA, it'd be easy, right? You would just say, oh, I see a C here. I'm going to add a G. I see a G here. I'm going to add a C. I see a G here. I'm going to add a C. I see an A. I'm going to add a T. I see a T. A. I'm going to add a T. Okay? It'd be pretty easy to build new DNA, so you'd think that an enzyme should be able to do this right off the bat. DNA polymerase that builds DNA can't start building new DNA from scratch. So at every single origin site, there is what we call a primer that's built by an enzyme called primase. Let's go back up. We'll fill in this blank. Primase is an enzyme that synthesizes RNA. Oh, what? Primase is an enzyme that synthesizes a short RNA chain called a primer. So write in your notes, a short RNA chain called a primer, and the new DNA strand will grow from this primer. What on earth does that mean? Well, here's what it means. I'm going to draw the primer in red. There is an enzyme called primase, and I'm going to build it in, make it like a little blob. Primase that has to build a short primer of RNA molecules. Oh, RNA molecules. And it builds these RNA molecules in only one direction, in the 5' to 3' direction. So it lays down a short RNA sequence called a primer. The primer complements the DNA. So when there's a C, it puts in a G. When there's a G, it puts in a C. When there's a C, what was that? A G, it puts in a C. When there's an A, it puts in a T. Oh, wait a minute. It doesn't put in T's. It's RNA. It would put in a U. Okay. A short, maybe 10, 12 nucleotides long, a short primer made of RNA has to be laid down before we can build DNA. This seems really strange, but that's what happens. And I had to make this clear that the primer has to be built in one direction. Primase only builds new DNA in the 5' to 3' direction. So it builds the G, the C, the C, the U, the A, etc. and it only works in one direction. It starts on the 5' end and builds on the 3' end, or adds to the 3' end. So at every single origin site, a primer has to be laid down before new DNA can be made. It seems really awkward, but that's what happens. Let's draw in the blanks here. See if we can fill these in. The upstream of our replication bubble, we've got this molecule unwinding and relieving twist in DNA. That's topoisomerase. You can finish that. Topoisomerase. The helicase unwinds the parent DNA. The single-stranded binding proteins hold the parent DNA unwound, and primase, an enzyme here that's shown in red, builds a primer. And notice that our primer is being built in the 5' to 3' direction on that strand. Now we're finally ready to build new DNA. And what we use to build new DNA are DNA polymerases.